Hello, this is Brian Kinghorn, and I'm going to introduce you to optimizing the index and using Matesel to generate mating designs in our rather successful bean project funded by ACIAR. So, as with anything in life, there are two key questions. One is where to go, and the other is how to get there. And the program Desire I'll talk about is all about where to go. Matesel is largely about how to get there, but it also helps you with the question of where to go. So I'm going to start with the old program GenUp for Windows, which we use for teaching in some parts of the world. And this is a module called Ellipse, which looks at the traits in our bean project, yield, cooking time, iron and zinc, and only looks at two of those at a time in order to discover where we could go. So we start right here in the middle, and after a certain period of time we could have increased yield with no change in cooking time, or in fact been anywhere on this yellow ellipse, which tells us the extent of how much response we can make in those two traits. And this is just a small component of Desire. Desire is now over here on the right, looking at all four traits. It's a bit more complex to look at, but it's the same sort of thing. And you can see that for cooking time and yield, we've got a more elongated shape of this ellipse. And that's because we've got more power to change cooking time, because this is multi-trait selection, not just two-trait selection, and information on the estimated breeding values of iron and zinc are increasing the ability, in particular, to change cooking time. And that's why this ellipse is less extreme than it is in the Desire example. I'll emulate the Desire example just by changing the genetic correlation. And there we can see that because we have a negative correlation between these two traits, we can make quite a lot of gain in positive direction for yield, at the same time negative direction for cooking time, which is highly variable. In fact, for all four traits, we are extremely lucky in this project for the types of changes that we can make to our traits. And that can be seen here in the responses that we have chosen in the Desire program, with a good increase in yield, almost maximal decrease in cooking time, and also almost maximal increases in iron and zinc. If, for example, we wanted cooking time to not change at all, so I can click here and say we didn't want any change in cooking time, and if I were to fix that, then we would find that in order to achieve that no change in cooking time, we would have to be within these red ellipses. So if I click anywhere here, I cannot get us outside these red ellipses, because I've constrained cooking time to no change. So that gives you a little bit of an idea of how we can use this program to discover what sort of outcomes we can have. But the decision has been very easy here. Just minimize cooking time, maximize iron and zinc, with some small compromise in yield. We could actually increase yield beyond what we have chosen it to be, but there would be some compromises elsewhere. I'll just finish this session with a look at a particular issue, and for this I'm just going to increase the genetic correlation, negative genetic correlation between cooking time and yield in order to make the point. Let's say that we were aiming to go to this point over here, which is not necessarily very sensible because it increases cooking time, but let's look at what that does. Here it is in the PowerPoint. The point I want to make is if we were specializing on two different lines, one going for higher yield and the other going for a higher cooking time, not a sensible thing, but we'll get to a sensible direction in a moment. If we made lines in these two directions and then crossed them, we would achieve a result just here. What that means is that there's a very small compromise in what we would have achieved if we'd gone in the consensus direction in the first place. So in this case, when you're working against a bad correlation, which this would be, you can't move so far in this direction, the upside in that is that you can diversify the directions so that you may have some good genetic resources for doing different things in the future and not make much compromise. However, if we look at our situation, where we generally want higher yield and lower cooking time, if we specialized in these two directions here and then decided to cross in order to make a consensus, we would end up at this point with a big compromise over where we would have been if we'd originally gone in a sensible direction. Now what we are doing in this project is going in that sensible direction. And the point of me showing you this is to be careful that it's not just where you can go, it's the flexibility that you have in order to use the genetic resources that you make once you've got there. Luckily in this project it's quite straightforward. Just go for the best values in these four traits. So I would just underline that what we've been looking at here is a strategic tool, and it's strategic in that it is telling us about economic weightings or implied economic weightings that we must use. That information is over here, so the implied economic weightings that we require to get the result on the screen are these values here. So we have to multiply each of the genomic EBVs by these values to get an overall index to use. So this is a strategic desired gains tool. Here it is with a different example, a bit more involved, showing the ellipse constraints that are made by fixing certain desires as you progress. But we can go for tactical desired gains, which is included in Matesel. Just to remind you that Matesel is basically about how to get there, but it does give you a chance to decide where to go. And with Matesel, there are very many different issues which are motivating plant breeders in order to have the outcomes they want. Matesel pulls all of those into a single framework. And here it is. We'll have a look at an example now. And the example is an earlier run done by Renu and Wallace on our bean project. So what you're looking at is the results that are generated earlier. And this is our frontier, which shows the overall progeny index across different traits. So that's the overall response that we expect to get across traits. And this is parental co-ancestry, which is a measure of how diverse the population is. And we want to be to the left with this, with less co-ancestry, more diversity. And we want to be high in progeny index. So we're aiming in this direction. And in particular, Renu has taken us to 50 degrees. 0, 10, 20, 30, 40, 50 degrees. Here we've got the inbreeding that's predicted in the progeny, and you'll see some changes as this run is progressing. We have the individual co-ancestry, it's the average relationship of each individual plant to the whole of the rest of the selected population. And here are the traits, yield, iron, zinc, cooking time, and the overall index. And so what I'm going to do now is just show you how this can be used as a tactical desired gains tool, because we're actually going to change the individual plants that are selected and how they're crossed in order to achieve what we want, rather than setting up rules, which is what the strategic approach does. So if we look at these distributions, the black tick is the average of the candidates, so that the response is the difference between that and the red tick, which is the predicted merit of the progeny of the crosses that are going to be given according to what this solution is. So this is for yield, iron, and so forth. If we look at cooking time, we can see we're going down in cooking time, whereas for all the other things we're going up, which is the desired direction for everything. Okay, let's just say for cooking time, if I click that, and I'll say we want tactical desired gains, and we want it to go to a particular value. If I click invoke, this line turns red, and if I resume, we'll see that the solution changes, so that the response we get is exactly on that line. And I can emphasize that by bringing that up a bit bigger. But you'll see that we compromise. We've got much less response on the progeny index scale, and indeed we've actually got more co-ancestry, so more inbreeding and less diversity in the population. So that wasn't a good move, but it does show you that we can aim at a particular outcome in terms of tactical desired gains, or we can do anything like set a minimum value at a boundary. Here we have that, or any of these other manipulations that we can make here. So these are tactical desired gains or tactical desired outcomes that we can achieve. Well, that's really all I want to show you, except to finish this off, let's print matings. And what that does, it says that the crossings that you'll make between this plant as a male and this plant as a female, etc., are shown on the screen now. There we are. And there may be more duplicate matings in there than you would like, but that's something which Reno has controlled in the results that were used for us in our project. Well, that's all I've got to talk to you about today, and thank you very much for listening. Open for discussion.